thank you all for coming. This is my presentation. Tetrapteris albitus is a white marlin. I think that's pretty cool. So these are quite an overexploited species. 1,000 to 2,000 metric tons are taken out of Atlantic waters each year. They are the smallest of the billfish, so they are closely related to such things as swordfish, sailfish, and the blue marlin, which inhabits the Pacific. They seem to be site-oriented daytime feeders. We don't know much about their life history or about their behavior. We do know that they're long-lived, solitary, they're pelagic predators. They have a very high metabolic rate, high growth, and fast development, which takes a very high caloric intake. And they are very important sport fish for the Florida and the Gulf Coast, as well as up toward the Atlantic seaboard. There's a lot of tourism dollars they bring in, so these are quite an economically and ecologically important species. So this map demonstrates their native range. It extends from about 45 degrees north to 35 degrees south on both sides of the tropical and subtropical Atlantic. They can be found in both coastal and offshore waters with about 100 meters or more deep, and they stay above the thermocline. So it would seem that their distribution is temperature limited. And so this was what we did to get our samples. They started out sending some grad students from our lab on a cruise to go look for tarpon larvae and eggs. We don't know much about the breeding habits of tarpon either. So this was going to be a big project. They took plankton and newston. For those of you who aren't fish nerds like me, plankton is the subsurface layer drifting in the water column, and newston is right at the surface. You can actually see right here, that's a newston net. They tow behind the boat at a specified speed, and all of the little samples go in here, and the net narrows down towards the end, where there's a specimen bottle that collects everything, and then we get to take it back to our lab. They were about 250 kilometers west of Tampa, in the northeast Gulf of Mexico, right here. And so that was a really cool experience for them. This was at the beginning of July in 2011. And we thought it was interesting to note that all of those samples that contained the juvenile marlin were found in the newston layer, so right at the very surface. We'll expand on that a bit later. So it turns out that where we found fish was in the Sargassum. Sargassum is an important pelagic habitat. It's a kind of seaweed and it floats, and it forms kind of a life raft for a lot of these different fish larvae, as well as invertebrates. We found filefish, flying fish, tuna, scombers, puffers, mackerel, and quite a few more. And unfortunately, no tarpon. But we found something really cool. 6 white marlin juveniles were found in the nets and pulled from the Sargassum. And now, white marlin are a sport fisherman's dream, because these are a rare and elusive species even at a meter or more in length when they start catching them. To get 6 of them at this age, my advisor was flipping out. He was so excited. He told me I was so lucky to get to see these, and it turns out we found some really cool stuff, so that was exciting. They range from 6 to 30 millimeters in length, and so we figured they were between 1 and 2 weeks old. These guys are non-synchronous spawners, so you're not going to get the entire population all together at once. So they're going to be slightly different ages, but there is still a distinct breeding season in that sort of late spring, early summer time. This is one of the guys we collected. He's about 28 millimeters, and all of those samples were preserved in formaldehyde. We were able to make a positive species ID on them because they have this unique cranial pigmentation. So our dissection methods were pretty simple. We put them in a petri dish under a thin layer of water and pulled them apart with dissection needle and tweezers. This is one of our unfortunate victims. We removed their stomach and gut contents because we were looking at what they were eating, because again, no one has ever looked at the larval biology of these fish before. We just don't see them. And so we removed their stomach, and we made indices to quantify the fullness and digestion levels. So we were able to measure all that. We looked at how much was in their gut, what proportion of their body length it was, and when they weren't so far digested that it was mush, we were able to look at exactly what they were eating, so that was exciting. And we also took a bunch of cool pictures. All of these samples were labeled and preserved in 70% ethanol. Out of 6 juvenile white marlin that we collected, we were able to examine 8 stomachs. And the smallest juveniles, well, of which we had 5, they ate copepods, and the larger ones, so that would be 25 millimeters and above, ate only fish larvae. And the running joke in my school is that bio majors aren't as good at math. I promise you, it's not because I can't count. What we actually found was that 2 out of the 3 larger juveniles actually ate other TL betas larvae. So they were cannibalizing each other. And I tell you, it was really cool when we opened them up and saw this fish head that looked exactly like, the fish itself. And we found that they were eating each other. This is the first recorded incidence in this species. Lipsky and Gorbanova, back in the 1970s, they found that sailfish and blue marlin also demonstrated this behavior. But nobody has ever really looked at it in white marlin before, which are, again, a rarer species. The prey ranged from 6 to 8 millimeters, and this was up to 25% of the total body length of the predator. So that's a pretty big gulp. The cannibalized white marlin were freshly ingested, and they were in pretty good condition. Again, we were able to use the cranial pigmentation to figure out what species they were. And because they were in such good condition, we were able to open them up and do a gut content analysis on the gut contents. And although the total numbers we caught were small, which would be why we're not really running a whole lot of numbers on them, we found some interesting things, like there are no day or night differences in feeding. We caught some of the samples in the morning and some in the evening, but we had equal numbers of empty and full stomachs for both of them, and the food that we found inside their stomachs was in widely varying stages of digestion. So you see, this kind of points to them feeding fairly continuously without a distinct time. So we're going to make a wild leap and draw the conclusion that the smaller ones below that 25 millimeter mark were zooplankivores. They ate mostly copepods, which are a microscopic crustacean that are often associated with these pelagic habitats. And the larger ones, they're called primary piscivores, meaning that they switch from microcrustaceans to fish prey at a very early age. And this is called size-specific predation. It's been found that even if you have the same total biomass of prey consumed, if you eat larger prey items, you'll grow faster, which is really interesting. So we think that these guys are switching over to fish larvae as quickly as they can because they want to grow and develop as quickly as possible. First of all, to avoid being eaten by ones that are a little bit bigger than them, and also because it's beneficial for them because they can travel more and search out prey more efficiently, seeing as they do undertake such long migrations. These guys have a very high metabolic rate because they are such strong, fast swimmers and they're fighting currents. So they have to have a very high caloric intake in order to maintain their metabolism. And this is really exciting. It is the first documentation of cannibalism in white marlin. It's really the first documentation of anything that they're eating at this rate. I've seen a few papers where they talk about what the adults are eating, and that's very interesting. But with fish stocks in decline all over the world and recruitment a big part of what contributes to adult populations, we think it's really important for us to know more about their larval biology. And then feeding occurs throughout the morning and evening. So these guys live close to the surface, and they're not very discriminatory in what they eat or when they eat it. Though we didn't find many of those copepods in the larger ones, so it looks like they're preferentially eating the largest prey that they can find. Again, 25% of the total body length, which points to them having a very good hunting instinct at a very young age, because if they're not going to be safe and effective in eating this, and they're not going to have a quick handling time, it wouldn't be efficient for their optimal foraging strategy for them to be eating this large prey if it was going to be so difficult. So these are consummate predators from the get-go. And then they are associated with the sargassum and require a high prey density for their growth and recruitment. Again, that's important for their management. And one thing that is a big issue in recent years is that there's a big industry that is harvesting sargassum and utilizing it for certain biocomponents for different industrial purposes. They want to expand this operation, and the argument is that sargassum can regenerate itself within 4 to 5 days, even if they take a few tons of it out of the water. And so it's not really going to make a huge impact on the ecosystem because they take out the sargassum. But what we found with the huge numbers of diversity of fish we found in our samples, with that big aggregation of such a rare species, to find 6 in that tiny area is amazing. As well as the fact that since this is an aggregation, it can maintain that high prey density. This is a critical fish habitat for these predators. And in addition to being important ecologically, there's also that big component of it bringing in a lot of tourist dollars. So if they're going to make the money argument, then we can say right back, well, you're going to impact this really important, valuable species that is quite rare, but clearly it's much less rare in the Sargassum habitat, and that is a crucial place for it when it is young, because they act almost as islands or life rafts in the big wide ocean with no cover, no protection. That's what the sargassum does for them. And so that's why it's such an important thing for them and with their early feeding habits. So this did bring up quite a few questions for further study, which we'd like to look into. My next project, hopefully, is experimental confirmation of the size-specific predation with some live specimens. We look at it in a more controlled setting and see what they're choosing to eat when we put in a different variety of size classes. And then we do some behavioral ecology work on that with optimal foraging theory and handling time. Development of a reliable method to age and sex juvenile billfish would be really nice, because that's just not something that was feasible for us to do. Even though the specimens were in very good condition, we were not able to figure out what sex they were, and there is some sexual dimorphism in the adults. The females do get larger, so they might grow faster, and then perhaps they would be preying on the males because they grow faster, but we don't know. So it'd be really interesting for us to be able to see what age and what sex they are, and that will give us a better idea of their life history and their feeding dynamics. And then the long-term study of prey selection and how that affects growth rates. Because the study I mentioned about eating the bigger prey to grow faster, even at the same biomass of intake, that's been done on different species, but not on this one. So I think that would be something that would be very cool to look into. I'm not sure if I'd be able to manage over the long term, because these animals have never been kept in captivity before. But that is something to look into. And I'd like to thank all of the people who made my research possible, and I had a great time doing it. Are there any questions? Yes?
How many stomachs do juveniles have?
Oh, they only have one. The joke was that we caught 6, and then we took the 2 smaller ones out of the bigger ones, and so that's where the extra 2 came from. They only have 1. Yes?
What's pelagic habitat?
Pelagic means open ocean, so away from coastal waters.
The sargassum you were collecting is in the Gulf?
Yes.
Is it? Okay.
It's throughout
Not out in the Sargassum Sea.
No, the Sargassum Sea has a lot of it, but it's a very, very common macrophyte in the ocean waters, and it occurs all throughout the
Right,
Atlantic.
so you find it in the Caribbean.
Yes, you will
The
find it
Gulf and everywhere.
all over the place, just floating in the water. A lot of aquatic organisms are very widespread like that.
Any other questions?
You mentioned aging the larva. Do you think you'd be able to get otoliths out of those?
We did look into otolith study, but the thing is, they're smaller than a grain of rice in a full adult that's a meter or more long. And so, as much as we tried, we could not see anything. Perhaps with a higher powered microscope and some fine tweezers we might have been able to, but it just unfortunately wasn't feasible for this size class.
I was going to mention scale or possibly spine cross-section.
Yeah,
Scale,
there is.
that may be a way to tell.
I read this really interesting study where they were using the anal spines and they were doing rings and looking at that, but they found that it wasn't very reliable, that they had some correlation but not enough to really make an accurate judgment. And also, as they get older, there's ossification of those rings, and so they become much harder to tell apart.
They'll reabsorb that the calcium in there, but at that age, they may
Yeah.
not, so that might be
So
something.
that would be something to perhaps look into with a higher powered microscope. That'd be very interesting. Thank you.
These sargassum, they form big rafts, or, I mean, if they're
Um,
taking out tons of it,
Huge
you
floating,
just trawl for small patches, or is it
huge
gigantic,
floating clumps.
like football field size? And
I'm not sure exactly, but I don't know enough about.
so the fish aren't just living in a small patch, is it? They're
No, no, and it's
the
not.
size of this room.
Yeah, I mean, it's not exactly, it's not super cohesive. It's a very sort of patchy habitat where they can go. But it's very clumpy. And so you're probably not going to, look down from a helicopter and see nothing but ground. But if you get closer, you'll see clumps here, there, and everywhere, and they just kind of go between those as far as we can tell.
Thank you, Carolyn.
All right, thank you.